So how do I switch to Chad? Okay. Chad Young. Thanks.
All right. So can you see my screen then?
Yes, I can see it well.
Great.
We are getting a little bit of a feedback, and from what I can see, it's only you and me that are not muted. So I will try to mute myself, see how this works.
Okay. I don't hear any of myself, so. But yeah, so Mario just asked me to do a quick presentation on just some of the things I've been doing. This is kind of just exploring the data. We're looking at the level 3 data, and one of the things that I wanted to do is come up with some kind of baseline that we can use to compare the different experiments against. And so, kind of semi-randomly, I picked a particular cell line and a particular gene to look at, the cell line A375 and the gene ERBB3, mainly because there's a lot of data for this particular combination. I was looking at the knockdown data in particular to see, is the knockdown data any good? Can we use it? And there was a lot for this combination, so I wanted to look at a baseline. So I went through and pulled all of the I guess distills that were listed as an untreated perturbation type. Came up with a little over 2,000 samples, and the first thing I did was plot just the expression level, kind of a density for all these experiments, and I ended up with a graph that looks like this, which is not what I expected. What I expected was, if this is an experimental setup where it should be the same across all the experiments, I'd hope to have one peak and then it falls off around there. But I see this secondary peak, and I wondered, where is this coming from? And when I was looking at the knockdown data, it generally fell to the left of the primary peak, but some of it was right on the secondary peak. So, is that knockdown data working or not? It's harder to tell because of these two peaks. And so what I did was I went back to the metadata and looked at various fields that are different between the untreated A375 experiments. And there were two in particular that jumped out at me as yielding very different profiles. The first is this perturbation description field. And so this density, as you can see, that secondary peak is coming basically all from this UNTRT untreated perturbation description, whereas the empty, and that's just what was in the field, you do get that single peak with a nice drop-off. And so one of my questions is, how is this happening, or what's the underlying cause? Because I don't know any particular difference between empty and untreated, and I was hoping maybe someone else had more insight. One of the things I thought of is there's different centers that maybe one was done on one set of equipment or different equipment or different centers that are producing this data. I really don't know why that should make a difference. But the other field I found that was pretty significantly different is bead batch. And again, I am a statistician. I do not know the particulars of how these experiments are done, but if you look at the bead batch, there's one particular batch, B7, that's really an outlier compared to the rest. And we have like batch B4 is that primary peak, and some of the other batches that weren't used very much are kind of spread out a bit, but it's B7 in particular that shows up as very different. And so yeah, I looked at a few other genes. This is another gene where again you see that difference. So it makes me question, does someone know why this is happening? Is there something underlying that's going on? Is something to do with the bead batches? Which ones are even correct? Should we be using just a subset of these experiments? These are all questions I've been thinking about, and I'd love to have any feedback on those. And so another thing that I noticed is there is truncation in the level 3 data at 15, and it's not a whole lot of the data, and for most genes it's none. They don't get that high, but there are a few genes that are very significantly truncated. There's one, in fact, for these 2,000 some experiments where 1,500 of them are right at 15. This slide says within 0.1, but I think it's actually within 0.01 of 15. But they're not all 15, and I don't know if there's something relevant about the fact that one is 15.002 and one's 14.95. But so those are kind of my questions, and I'd love any feedback or thoughts on if people know more about the biology or how these things were done. I'd love feedback.
So thanks, Chad. There are a couple of ways we can work on this now. I can unmute everybody, and you guys try to control your muting, and we see how it goes. And we can start like that. And then if that turns out that it's not working, then I will again mute everybody, and you will need to chat that you have a question, and I will unmute you one by one, all right? So let me try to unmute everybody first, if I know how to do that. So where is it? Unmuting thing. Oh, here. All right, so everybody who should be able to speak now, and please keep yourself muted unless, no? Unmute all. Unless you want to talk. So, unless you want to talk. So okay. All right. So we will try another way. Who was that? I will unmute Chad first of all. Okay, he's unmuted. And if you would like to say something, can you please tell me, and I will unmute you? In the meantime, I will ask Chad a question. So Chad, you first split the data by these two different labels, untreated and what was the other one? I forgot. And then when you were doing empty, right?
Mm-hmm.
And then when you were, oh, okay. So I have a couple of questions. Is this just a compound treatment data, compound perturbagens?
So I went in and pulled out everything where the perturbation type is listed as UNTRT, untreated. And that gives me this. And then within that data, conditional on what the perturbation description field said, I get this.
Okay, but I mean in terms of, so you basically use all data, 1.3 million columns to start
yes,
with?
I pulled out, it's 2,000, ends up being 2,040 or something like that. And that's what this is. I didn't do a density across the 1.3 million.
Right, but you 2,000 that you pulled, 2,000 columns. Say these were the criteria you used to pull them was just that they are. Oh, I'm sorry, I misunderstood. So these are not. These are the whole profiles are empty and untreated, right?
Yeah. There were 1,437.
Yes.
Where
Okay.
the perturbation description was empty.
Right. For the right. So this could be coming either from any type of perturbagen, right? So because empty sounds
no.
No.
These are all subsets of the untreated. They all have perturbation type of untreated, which is UNTRT.
Right.
I think it ended up being 3 different perturbation IDs.
Right, but they could be coming from the plate that had either chemical perturbagen or shRNA.
Correct.
Right. So I suspect that empty comes from either overexpression assay or the shRNA assay, right? While untreated comes from the chemical perturbagens.
Mm-hmm.
Is that reasonable? Does anybody else have anything to say about that? Nobody's.
Yeah, so the other thing that I was looking at is there's untreated, there's also control vehicle and control vector, and those aren't included in this.
Right.
Yeah.
So the control vector again would be part of shRNA most likely.
Right.
And control treatment would be DMSO basically. It would be chemical perturbagen treatment. Mm-hmm. All right. So now when you further subset these by bead batch, do you don't anymore condition on the first label, right? On whether it's untreated or
no, it is still by untreated. I was just looking at the untreated and saying, why is this having 2 peaks?
So these are now all untreated.
Correct.
Oh, I got you. And it's that. Mm-hmm. So within the untreated, you have this weird. No, you have a. I need to see both of them at the same time.
Yeah.
Okay, so untreated actually has the bimodal distribution. Most of it is low, but then some of them are in the right peak, right? At the high. And then you can actually further split this by bead batch.
Right.
But it looks like the bead batch and this other, what is it, untreated versus what is it, empty, that seems to be correlated, is it not?
Yeah. It is, and so I'm not sure, what is driving it, whether it's, bead batch, all the untreated happen to be bead batch 7.
So where are these coming? Is that from the overexpression or knockdown or something? Like Mario said.
So,
what are these batches divided by?
I didn't look at the other experiments that were on the same plate as these to say, this was like a plate of knockdown. But these are supposed to be, I believe, the controls across kind of all the plates. I didn't look to see, all the untreated ones. I didn't think about that. All the untreated ones happen to be on plates where they're doing shRNA experiments or something like that.
Okay, so I think I misunderstood. And so when you condition by these bead batches, they're still you just partition the nice peak, the big peak. The untreated ones are still there in one batch.
Right. So all of these have a perturbation type of untreated.
Oh, they do. Okay. All right.
Yes.
All
it's
right.
just that perturbation description field is different for some of them.
I got you. Right. But right. Okay. So they're all untreated, but here are just perturbation description for these. These are all both kinds, right? Empty and untreated.
Correct.
Okay. So my read on this would be that there was maybe this one batch of beads that didn't work well for this particular gene, and I don't want to put too much weight on this assessment. So if somebody else has any other idea now.
Yeah, and I mean the expression level by themselves are not that meaningful anyway, right? You would always need to compare it to something. So maybe this whole batch is just lower. So everything is lower, even the treated ones. So then it wouldn't really matter.
Yeah.
So anybody else has any comment there? Avi, is somebody from your group have any experience in this kind of QC? So let me just, if you guys could introduce yourself. So the previous person that was speaking, I believe it was Johannes Freudenberg. Is that correct?
Yes, sorry, I should have said that.
Okay.
Yes, Johannes from GSK.
All right.
Yeah, I don't think we look too much on that level of data. Or maybe, I don't know, I think maybe Neil or Chao-Nan can talk about their experience, but there's also some other people that I know from Colombia that looked at this. And probably Arvind can also say something about it. So I can ask him to join us now, or we can take that question to them and then get through email, like follow up on with emails and then figure it out.
So one of the things that we can do today, and this could be one of the case studies or questions that we can email Arvind up front before his presentation on Tuesday, right? So we can collect specific topics that we would like Arvind to address.
Yeah, I think it's a good idea. Because it's next week, so just now we have to raise the question. We can afford to, we can send him, maybe he can answer by email and then discuss next week.
Okay, that would be good.
But if Chao-Nan or Neil has some ideas, then please feel free to chime in.
So let's see if I, by any chance, have muted them for no reason. Yeah, there are quite a few people that unmute all again. Okay, so everybody should be unmuted now.
Chaunan's trying to get on the call. I don't know if you can hear me.
Yes.
Okay. Yeah. Anyway, this is Nicholas from Avi.
Okay, so there are, I guess, there are several questions here. One is what is the exact meaning of these two particular labels, and I think my reading is that one comes from untreated, comes from chemical perturbagens, and the empty comes from shRNA.
Okay.
Or overexpression. And then the other question is this one bead batch that behaves that way. So the third question is basically what Johannes suggested, that perhaps there was a shift in overall distribution but I would find this on the plates that have these measurements. I would find this a little more difficult to believe because that would be correlated so strongly with this one bead batch and the fact that these are normalized, Q normalized.
Right.
I would think that that's something that's specific to this batch of beads.
Yeah, and when I'm doing further analysis, I am comparing on a plate level, so it's a differential rather than looking at it compared to the kind of the whole profile.
Mm-hmm. But this would still introduce the noise, right? Because probably the relative expression level of these genes would be very different with respect to other genes on these plates.
Right.
Right? So if you were to pull information across, that would probably not work.
Can you guys hear me? Hi, this is Stefan. Not sure if the audio control works, but isn't the timing, I mean, if one is knockdown shRNA, one is compounds, and isn't the timing different? Kind of, which it could mean that there's different, perhaps a different expression level. And I don't know if this would be random or if they would be triggered by something, but maybe.
Right. I looked at some other things, and one was the time index, and most of the ones at this large peak are what the longest one, 96 hours, I think. And the lower times are also more distributed. So yeah, that'd be something I can definitely check, is to see, what's happening on the plates that have used bead batch 7 that might be different.
Okay, so I guess I don't hear many more suggestions at this point. So one take-home message is that this kind of QCs should probably be performed for all genes. The other is that we should add this to our battery of QCs and performance benchmarks, maybe. That is something that I was hoping that we can start working on, is the set of suggested benchmarks that we could use for signatures that we derive, but this will also depend on what exactly we are doing with this data. So if nobody has anything more to say about this particular problem, we can all maybe try to figure things out and share what we come up with. We will ask Arvind about this and maybe ask him to also chat about this next Tuesday.
I have a question. This is Peter.
Yes.
Hello?
Yes.
Yeah, so this is Peter from Beck. So it's, the bimodal distribution looks to me like, I mean, how do you explain that? It looks to me like you have one peak at the expected position, you know, but a much lower expression level, right?
Right.
So,
well, I guess.
Something different with the flow?
Well,
there's something different.
The offered explanation, I think, is that there is something different with this bead batch B7, that perhaps the probe or used on this particular bead batch works slightly differently than the other ones.
Would that be specific then for ERBB3?
That's my guess, but we can. No.
This is another gene that I looked at.
Okay.
And you have the same difference between those, you know, the two main batches.
Okay, so I guess I will change my story for 180 degrees. It looks like that maybe the whole batch is slightly different for all genes. Okay, well, let's ponder this offline, ask Arvind, and then maybe we can reconvene and talk more about this. So going along the line, so Avi, do you think there is still time for you guys to present on ENCODE?
Yeah, I think so. I think it's a pretty short presentation by Jan.
Okay, great. So we'll hear a presentation on processing ENCODE data, and I think this is starting to go in the direction of how do we actually meaningfully benchmark signatures that are going to be derived from these from these data, or at least I think we can cast this presentation. That's one of the directions that we can cast this presentation. So Avi, you wanna introduce presentation, or who should I switch the presenter roles to?
No, yeah, you should switch to Jan Ko. And I can just say that Jan has processed the ENCODE data in the past, and she recently upgraded or refreshed her analysis of the same resource, and we've been using that resource to benchmark various methods, computational methods for extracting signatures from gene expression data, and it's sort of like perfect for benchmarking the L1000 knockdown followed by expression and overexpression data sets, and also for building those transcriptional regulatory networks that Ziv and Kai are interested in developing. So I think it's very relevant, even though it's not links data processing. It's data that fits very well with the L1000 data and can be used to benchmark.
Hello? Can you hear me? Is it clear?
You're pretty low.
Yeah.
So if you can increase your volume or speak louder.
Hello? Is it clear? Can you hear me?
I think it's better. It's still a little low, but I can hear you pretty clearly.
What about now? Is it better?
Yes. I think.
Okay, great.
Thanks.
All right, so I'm gonna talk about the ENCODE data and what I've done with the ENCODE data. So first, for those of you who are not familiar with ENCODE, it is an international consortium founded by the National Human Genome Research Institute. There are many collaborators here, the list of the main collaborators, and also the aim of this project is to build a comprehensive atlas for the functional elements in the human genome. And the functional elements that ENCODE looks at are includes the hypersensitive sites, meaning the open chromatin sites, and also the DNA methylation, his modifications, and transcription factor binding sites, and also the RNA polymerase II occupancies. And recently they added the long-range interactions between chromosomes, as well as the enhancers, repressors, silencers, and insulators. Here is a cartoon showing the data ENCODE has collected. It is basically organized into cell lines and the assays that have done with the cell lines. ENCODE has the initial phase of ENCODE characterized 3 cell lines, and then they expanded gradually over the years, and now there are almost 400 different types of cell lines. And for each cell line, they have collected different types of functional elements, and there are also a whole bunch of ChIP-seq experiments for each cell type. This is a timeline for the ENCODE project. Starting from 2003 until 2007 is the pilot phase. There are 3 cell types, and they annotated 1% of the genome. The technology they use are mostly array-based. Starting from 2007, it's the production phase. They mostly adopted the next-generation sequencing technology and expanded the cell types to 147. And at the end of this phase, they claim that 80% of the functional portion of human genome has been annotated, and the data they collected are looking at over 2,000 protein coding genes and over 18,000 RNA genes. Here are some numbers. So the data produced from the pilot and production phase are hosted as a UCSC genome browser. Starting from 2012, they expand the production phase. They now there are more than 300 cell types, and also there are almost 300 antibodies. And they also started looking at some new functional elements such as the RNA binding proteins, silencers, enhancers, and the long-range chromosome interactions. And the new batch of data are now hosted at the ENCODEproject.org website. So here is a screenshot of the UCSC genome browser. I think I better switch to the website. Okay. So here is the, oh sorry, not this one. Yeah, this one. Here is the ENCODE data at the UCSC genome browser. You can basically browse the data from a data matrix. It's organized at cell type level and also the assays. And there is extended data matrices for the ChIP-seq data set. To download the data from this UCSC browser, you can go to the download link here, and then they categorize the data into different genome build, and also here you can see different assays. The problem with this website is that first you can only access data through their FTP links, and also the metadata is not very clear. Yeah, so for example, you can get the metadata from their cell type or antibody list, but it's not very specific for each individual data set. And when I process this batch of data, I basically parse the file name of the file names, and then get the information mostly from the file names, such as the assays and the replicates, etc. Yeah, something like this. And as for the new website, ENCODEproject.org, it has a lot more functionality for us. So let me show you in the browser. Oh, it's not this one. Yeah. So this is the new ENCODE data website. It gives more filtering functions for users to select from. For example, we're interested in a ChIP-seq data set, and we can choose different organisms, organs, and treatment, and also the data types. So, and also when you click on a specific data set, if you're a regular user, it gives you a well-organized summarization of the metadata. And if you want to see more information, you can look at in the JSON format, where there are a lot more information associated with each experiment. Okay. And also, the data hosted at ENCODEproject.org can be automatically accessed through the REST API. The metadata are organized in JSON format. So this is a data model example of their data model, and you can find the full schema on the GitHub site. So here is an example of what I've done with the ChIP-seq data from ENCODE. So there are a few steps. First, I download the ChIP-seq peak files and also the metadata. So ChIP-seq, the raw data for ChIP-seq are just the FASTQ files, and then you map these reads to the reference genome and then identify the significant peaks. So I download the peak files and also match each peak file to their metadata. Now I know the cell types and the transcription factors or his modifications.
Technology available.
What is it?
Whoops.
Hello? Hello?
Hello.
Oh, sorry. Okay. So after I match the peak files to metadata, these peak files are in the BET format, meaning that for each peak there is a chromosome coordinates. And then I take these coordinates and map them to genes, meaning that I look for the genes that are close to this peak. And then I generate gene list for each of the experiment. I did the most of the steps using in-house scripts in Python and Java. And here are some numbers for the data set. So for the new batch of ChIP-seq data, we have both human and mouse samples. And we have over 600 hit modifications, as well as some CTCF and RNA fold2 occupancy. So this is how it looks like after I generate the gene list for each experiment. So this is just an example. This is actually the ChIP-seq data from the pilot and production phase of ENCODE project. So basically, this is the first column is a label for the gene list, and it is the transcription factor in the. So this is the transcription factor, this is the cell type, and this is a list of genes that are associated with the peaks from the ChIP-seq experiments. Here is a summary of the data sets we have. So these data can be accessed through Enricher. It's a web app developed in the lab. So far, we have the number of experiments are the data set we got from ENCODE, and then after I merged the replicates, unified experiments, there are this many terms for each of the data sets. This is for the transcription factor binding sites. This is for his modifications. We also have some additional his modification data from the epigenomics roadmap project. And we also have some ChIP-seq data from CHIA, which is a manually curated database. And then we will add these new batch of data soon. So here is an example of how to use these data from ENCODE. So here is a figure from a study we did last year. We collect all these ChIP-seq data, and then we create a network consist of transcription factors and his modifications. We also connect them through the his modifying enzymes. So we would like to take these transcription regulome and start to look at the gene expression data from the L1000 experiments and try to identify the upstream regulators for the up and down genes. And that's it. Thank you.
So I think just to add to what you can do with the ENCODE sort of like process data, you can start validating L1000 transcription factor knockdown and overexpression by looking at the differentially expressed genes when you knock down or overexpress the transcription factor should match somehow with the transcription factors that were profiled by ENCODE. And the more overlap you see, the more likely your methods to extract the differentially expressed genes and sort of like normalize the data, the better. So that means that, and if you do it in a high throughput, then you can actually see a pattern of like how well the L1000 captures the biology and the right genes are changing when you process that data.
Right. Yeah.
Okay.
Other questions, comments?
Hi, this is Jarek speaking here on Mariusz side. I was just wondering about overlap between cell lines in LINX and in ENCODE project. Can you comment on that?
So as far as I know, there are not many overlaps between the tier 1 and tier 2 cell lines with L1000, but you can browse the cell types on their website here. Oh, sorry, this is mouse. There is a full list of all the cell types included in the ENCODE project. Yeah, so you can find overlaps from here.
So I'm curious, did you had pulled out some targets right before
Yeah.
for these? You know, we've done similar analysis, and then it turns out that if you look, at least in our hands, if you look at dominant targets for like more than half of ChIP-seq experiments there. They are all turn out to be proliferation-related genes, like cell cycle and that kind of stuff.
Okay.
And was that your experience as well?
Are you looking at all cell types? Are you looking at transcription factors or his modifications?
Yeah, well, it's basically transcription factors in any kind of setting. So it's just transcription factor ChIP-seq. And then if you kind of look at, top, I don't know how many genes for each transcription factor, it turns out that vast majority of transcription factor have huge, vast majority, more than half of them, have a big overlap. And all of the genes that are in the overlap are cell cycle and proliferation related.
So the data I have been looking at, meaning the data from the pilot and production phase, I didn't observe that most genes are related to proliferation. I think maybe the cell types matters because the first two appears there are many cancer cell lines. Maybe that would explain. But
so I'm sorry, did you say that you do see it or you don't see it?
I don't see that.
Oh, you didn't see it. Okay, no, it was just, yeah. Well, it's going to be interesting to compare once we have both things out.
Yeah, that would be interesting. You can also get the data from the process data from the Enricher website. Yeah.
Also in your
exactly where it is.
In your CHIA2 paper, there's some other figures that show the overlap between all of the samples. Like for example, this, I think it's this one, and you can see that they don't necessarily 50% overlap. The gene lists that Jan got, they don't necessarily overlap like 50% of them like you described. So maybe we should exchange notes to see what happened.
Yeah, definitely.
Yeah, that would be good.
Okay, not that one.
Yeah, I think in this
I can also send you the papers that include the analysis I did with this data. Maybe there's something.
Oh no, I have CHIA-PET paper, and it was on our in our list for a while now to compare what we have with what you guys have. I was just trying.
This figure is all of the experiments comparing. The rows and the columns are the transcription factors and the histone modifications, and you can see that there's some clusters that are highly overlap, but overall it's pretty sparse.
Yeah.
And so it's you get very different gene sets, and I'm pretty sure that they're not going to be all like cell cycle.
All right. So, but okay, I see, or maybe I overstated what a little bit what we had, but there are two big squares there, right?
Yes.
Yeah. Okay. Okay, great. So I think this is a great first step. Once we start producing different kind of signatures, we can correlate these directly with shRNA and overexpression perturbations, but we can also compare for many drugs. There are some putative targets, and we can also see if these are represented in downstream activity of the transcription factor that you expect would be mediating this drug effect.
Yeah.
Okay, any other comments, guys? Does anybody want to mention anything else or talk about anything else today?
I have a question. This is Peter. I was going on the Enricher site and then clicking on, say, data set statistics for the kinase perturbations from L1000. Okay, that takes me to the Lynx cloud. It doesn't take me directly, as far as I can see, to the data set that's in question, does it?
No, it's not available.
It's not available.
I mean, we have it. We can send it to you, but it's not available for download yet.
Okay, fine. So it's just that I'm not missing something or just being myopic.
Yeah, Jan misspoke. It's not. We haven't provided all the data for download.
Okay, good. And secondly, I have a question about next week or the 2 weeks, whether or not, I mean, the data that Stephen and Clarissa have been working on is not really L1000 relevant at the moment. I mean, they've been working more on the disease data set rather than on the cell line data sets. So I didn't know if it would be appropriate for Stephen to talk about the approach he's using or what.
So, Peter, is it still gene expression data, or it's phosphoproteomics data, or?
This is a proteomic data. This is a phospho. He's actually analyzing all of the phosphorylation, the acetylation, the methylation together, and trying to look at correlations upwards or downwards between all of those data types. But I didn't know if I don't know where he's at, if he's ready to present, but I didn't know if also if it's relevant, because we don't really even have the P100 data points that I would like to get a hold of just from Jacob, from his data. So we in fact could interrogate this data with the, say, P100 data, the phosphorylation sites. I mean, that would be relevant possibly, but I don't think we're ready to analyze it or discuss it in context of either the P100 or the L1000 data.
So, I mean, I see the value if Stephen would or you would want to present a little bit about what you guys are doing, because eventually the objective is to use all available data and to look for connections between different data types. So that would be interesting. I'm curious, why is there any roadblock between you and P100 at this point that you cannot get the data?
Just the roadblock, frankly, is time.
Oh, okay.
I've been incredibly overbooked up until last Friday, and so I haven't had a chance to go back. I hope there isn't. I mean, it hasn't been obvious when I've tried it, but I did get some information from the Broad that Supermanian that might be helpful to get me in, but I haven't really investigated that further.
Okay, so
if there is a problem, should I contact? Maybe I could contact, I guess, Jacob or what?
Yeah, I mean, I think there should be no problems if you contact him directly, or you go through usual channel, through Lynx Cloud. If you run into any troubles, then let us know. And but
we supposedly have we presumably have access. Aravind sent us. I sent the name of the people here, and I think we're supposed to have access.
Okay, so let's speaking now about 2 weeks from now. I think it would be interesting to hear, in general, at least maybe a short presentation. It doesn't have to be the whole period, about what you guys are doing, so that people can start maybe making connections and think about your way of analysis as well.
That would be, I think, fine. I think Stephen or Clarissa would probably be very excited about giving the overall view of what we're doing. And I just didn't want to interpose something that's inappropriate for the L1000 consideration, nor break anyone's great concentration on trying to understand the L1000.
So we'll continue discussing L1000. Eventually, we do want to incorporate other data types and talk about other data. So I think having a piece of that already is a good idea.
A prelude, so to speak.
All right. Okay. Is there any other suggestion, comment for this week's meeting? Please send. I would like to try to collect some specific questions for Arvind to focus his presentation, because he has a collection of hundreds of slides that he can pull out specific slides from to address our specific questions. So I think it would be good to direct his attention to something specific. My specific question would be along the line of defining gold signatures. These are the signatures that they believe are good and strongly reproducible and so on. We can toss in maybe some comments on Chad's analysis and what happened there and what is his thought on it. And then one suggestion was from Roger to maybe discuss a little bit about uses for this data in terms of what scientific questions he thinks can be answered by this data. But please, if you have any thoughts on what else you would like him to discuss, let me know.
Peter, again, I have a question. So I sort of gauged the whole world and my whole schedule by Google Calendar, and it would be terrific if invitations were sent out for our meetings on Google to our email addresses, which are directly linked to Google Calendar.
If you send me the okay.
Yeah, our email address.
Yeah. So the same address, just send an invitation. Okay. Well, yeah, I'll try.
Correct.
To do that.
For Clarissa, for Stefan, for Stephen, for myself, for Sean, it'll all go directly to an invitation on the calendar.
Okay. I'll see what we can do
That would
about
be
it.
terrific.
Yeah, we also have a DCIC calendar, that a Google Calendar that Stefan already set up. So we should add you to that to make sure
Correct.
that you
Yes, that would be great. Excellent.
And we should.
And I
put
think
the
Stefan was going to send us wiki access. I'm not sure that we got that.
Yeah, we need to make sure that we get it.
Okay, good.
Let me check, okay? I can add you guys to the wikis and to all the documents and everything.
On the calendar.
Excellent. Thank you so much.
Hey, Mario, are you going to share the video of today's meeting?
Yes, I will. So it's slightly different process. I have to first convert it this time, and then I will upload it and share it. So probably tomorrow we should have I should have a link, and I will send it out.
Okay, great.
All right. Well, thanks all. I hope I talk to you next week.
All right.
Excellent.
Bye-bye.
2 weeks.
No, next week. Next week. Next week is Arvind talking, and then in 2 weeks we have other topics, right?
Okay, great. Excellent. Same time, same station.
Yes, same time, same station.
Bye.
Thank you.
Bye.